good interactive session from your end also. So a very warm welcome to all the participants out there. And I'm really happy to interact with all of you. And I would really wish that you take home some lessons today, and this will be of a very good benefit to all of you, right? So just give me a second. Let me just share my screen. Okay, so there we go. With the talk, the first thing that should come to everybody's mind is, why should we learn plant developmental biology, right? Why are we here to learn this particular topic? Why is development in plants so important? So when you look at a hibiscus flower, which is red in color, as a child, I always used to be fascinated and think that hibiscus is red in color. And this was the concept that we always carried, till I found another one, which was having white colored petals. After some more time, I started looking at hibiscus flowers with pink colored petals. And I always used to wonder why there is so much of a difference in the coloration of these petals. Now, this is about a single flower, but when I looked at other flowers, I found that Vinca rosea, it is having a different architecture of the flower compared to hibiscus. There are some flowers in which I count 5 petals. There are some others in which I count 6 petals. There are some in which I cannot count petals. So that is because the flower is not actually having petals. Instead, it is made up of something which is called ray florets and disc florets. So I used to wonder, where are the petals, right? So it is extremely important to learn developmental biology, because this topic would help you gain an insight into how development takes place in plants, how there is morphogenesis and how there is differentiation in plants. It is important to know what is the radial patterning and axial patterning in plants. And plants, believe me, they present a diverse array of genes and gene interactions, which actually let you understand why a particular flower has a red petal, why some others have 5 petals and 6 petals, so on and so forth. So this is just one example that I gave. Apart from that, when you look at plants, some of them are short, some of them are herbaceous, some of them are shrubs, while some of them are trees. There are some plants which we need to water every day, but there are some plants in nature which you do not ever, provide water, but they still are sturdy. They grow so well. So why is that so? So this is because of the difference in the developmental patterns of these plants. So it should be very, very fascinating for all of us here to understand why and how development takes place in plants. Now, developmental biology is a part which is included in your CSIR-NET syllabus. And as Sir rightly pointed out, there are at least 1 or 2 questions which are put up every year. And another thing is that developmental biology, you are simultaneously going to learn in case of animals also. So in today's lecture, let us have a quick peek into what are the differences in the developmental patterns of plants if you compare it with the development in animals. So going ahead with the talk, the topics that we're going to cover today are broadly the overview of plant development. Now, developmental aspects are something which you have already been studying in your graduation curriculum as well as in your post-graduation curriculum, okay? But what are we supposed to know here is, what are the integrate mechanisms and in-depth aspects of development. So first, I'll take you to an overview of what plant development is all about. And then the next topic that we're going to talk about today would be the individual development of root, stem, and leaves. In this, we are going to cover What are the different aspects of meristem development? So plants have a tendency to undergo differentiation for a longer period of time. And that is by means of the meristems that they carry. So how are the meristems going to be specified in case of root, stem, and leaves? That is what we are going to discuss at length. The next topic that we are going to talk about today would be about embryogenesis and patterning. So just like in the animal development, in plants also we have embryogenesis and patterning systems. So what are these systems? How do they operate? This is all that we are going to talk about today. This would be followed by an intense discussion on transition to flowering. This is an extremely important topic for all of you from your examination point of view, as well as for your understanding, because we have been knowing that plants, okay, they flower, like they undergo vegetative phase and then they undergo a flowering phase. But when they transit from the vegetative to the flowering or the reproductive phase, there are lots and lots of changes that take place in plants. And there are lots and lots of gene clusters which actually help them to transit to flowering. So what are these aspects? That is also something which we will see. And this would be followed by the last topic, which would be on floral development. Floral development is a topic which has been extensively studied in plants, but all the studies, however, had always focused on Arabidopsis. So today, just to give you a little different view, I would like to focus a little bit on the antirrhinum plant as well. So antirrhinum, which is commonly known as the Snapdragon. Okay, so with this, let me move further. Right. Now, plants adopt specific strategies when it comes to their development. What are these strategies? The first and the foremost important point in terms of plant development, which makes them so different, is that the life cycle of all land plants includes two phases, that is the diploid phase as well as the haploid multicellular stage. And because of the plants actually displaying such a property, they are believed to have a haplo-diplontic life cycle. What is this is something which I would explain you in the next slide, okay? And intermittently, we would also be solving some questions which would get you a better insight in, and let me also know whether you're understanding it or not. So this is the first form and important aspect of plant development. The second thing is that the germ cells in plants are not set aside early in the developmental stages. Now, when I say this, what do I mean? So even before I tell you what it means, I would first like to have some interaction from your side. What do you mean by germ cells? Can anybody answer this question? You could just post it in the chat boxes. Yes, anybody? Okay, cells which can give rise to gametes. Okay, they generate gametes. Cells from which gametes develop, and germ cells are those cells which give rise to gametes. Okay, okay. Now, what is so special about germ cells? And what is the difference in the germ cells in plants and animals? And when we are using this particular statement, what do we mean to say? Yes, we are having very, very quick answers. That is really, really nice to see that you all are so responsive. Good. Yes. Now, when we talk about the development in animals, the most important thing is that the germ cells are determined at a very early stage as to what is the pattern of development that they're going to undergo. But how are plants different? So we are saying that they are not set aside very early in their development. So in case of plants, what happens is that these germ cells, they tend to give rise to more amount of, they tend to arise from the somatic cells in the adult. And a very typical example of this is the floral meristem, which is seen in flowering plants. So when plants undergo development, they are in a continuous state of development. It is not that at a particular point of their life cycle, they just stop developing, okay? Because in human beings, in animals, there is a particular pattern of growth and development, both. But when you talk about plants, this is a continuous process. And this takes place at many levels, okay? So these germ cells, instead of being predetermined or instead of being committed at a very early stage of development, they can actually arise from somatic cells at a later point in their life cycle. And this is what happens when they transit from their vegetative phase to the flowering phase. How, why, when, and where. This is something which we will discuss in the later part of the presentation, okay? Then another aspect is that plants show an extended morphogenesis. So here I would like to know, what is morphogenesis? What is your understanding of morphogenesis? I must say people are really working out and finding answers. So very, very good thing to see. Yes, we have the response. Formation of a structure. Exactly what structure? External appearance in Genesis means generation. Correct. Very good. Ria. How plants look like? A show. Very good. Different parts of plant. Who's this? Through a journey. Very good. That's a more specific answer. So formation of different parts of plant, root, stem, leaves, flower. Okay. Thank you very much for being so cooperative and so attentive, right? Okay. So morphogenesis is basically, Yajurva has also answered, Kinjal and Yajurva. Very good, very good. I'm seeing some very good answers and they are not repetitive. That is very good. Okay, so morphogenesis is a phenomenon which talks about, formation of different organs. When I talk about plants and when I say plant organs, I am talking about the formation of root, the formation of shoot, leaves, stem, flower, fruit, everything, right? Now, when I say that plants, they have an extended morphogenesis, I'm actually trying to tell that they can adapt to the changing environmental conditions. They are not those kind of organisms which can move from one place to the other. But they, at the same time, are those species which could actually adjust and acclimatize themselves to the change in environmental conditions. And their organs, they also undergo a differential development when it comes to an availability of light, for example, an availability of photoperiod, when they grow in the dark. There are so many parameters where these plants are wonderful beings which can actually acclimatize and adapt themselves. So their morphological structures, when they develop, this is like they take into account all of these conditions and they actually modify themselves to suit to the external environment. So don't you think it's fantastic? Another important property. So the next thing is that they have developmental plasticity. Now, this is one more thing which I would like you all to answer. What do you mean by plasticity or developmental plasticity? You guys are really quick. Adaptability. It can adapt themselves according to the environmental conditions or habitat. Okay. 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 So that is answered by Mansi. Yes, Mansi is right. Kinjal is right. Ria is also right. Very good. So you have very good basic knowledge about development. Now, this is where an organism actually is adaptable to anything. When we feel extreme heat, when we feel extreme cold, okay, we try to either move away from that condition or we just try to take, some precautions against that condition. But what plants do is really remarkable. They undergo changes to suit to that environment. Okay, so all the morphological structures that are formed during plant development is actually coming in as a result of this plasticity that they possess. This plasticity is not just simply supported by root developing into the primary, secondary, tertiary. This is something which is not only a characteristic feature of the vegetative development of plants, but it is also there up to the reproductive stage of the plants. So this is something which makes them much more flexible. I'll come to that point later on also. Then, independent evolution of developmental mechanisms. This is a point which has been debated since ages. There are lots and lots of theories which are pertaining to evolution, and there are theories which state about the common ancestry of plants and animals being, formed from the same lineage. So here what we are saying is an independent evolution. The studies on independent evolution on plants has actually come in from the fact that the homeotic genes which they possess, they are a different set of genes when you compare it with the ones that are studied in the animal systems. So here again, I have a question. What do you mean by homeotic genes? It has everything to do with the topic that we are studying today. I would like the others also to answer. Come on, it's so simple. I mean, if you're wrong, it's okay. Developmental genes which define a region in the organism, from Mohit. Very good, Mohit. Can we be a little more specific? Genes which can regulate the anatomical features. Mansi. Good, Mansi. Okay. Okay, so at this particular point, I don't see much of the people participating. Oh yeah, regulating the development of anatomical structure in various organisms. So when there is development in terms of organ differentiation and organ patterning, that is basically under the control of some genes. And the genes which are going to control this particular parameter in plants as well as animals, they are known as the homeotic genes. So when we are saying that there's an independent evolution of developmental mechanisms when it comes to plants and animals, and that plants are actually independent, there is a set of genes, there's a homeotic gene cluster, which is called the Mads box genes. As a student of MSc, or if you're pursuing MSc, or if you have already cleared MSc, you might have come across the term called the Mads box. I'm going to elaborate on it in the last topic where we are going to talk about floral development. But this is a set of genes which was found in the primitive animals as well as in the primitive plants. But plants still have them, and these genes are still actually specifying the developmental patterns in plants. So the character or the fact rather that the plants are still possessing these genes makes us believe and give an evidence that they have an independent evolution of all of the developmental mechanisms. But this is not a statement which could be given only on the basis of one factor. We have to have a quick look at other factors also, right? So this is basically the gist of what are the strategies that the plants adopt for actually their developmental patterns. Okay, now I'll move ahead with the topic and let us talk about the haplo-diplontic life cycle. That is just talking about the plants having both of these phases. So here, if you look at the life cycle, you will first of all see that there's a haploid. You can see my cursor? Okay. So there's a haploid generation and there's a diploid generation. And if you look at the way it is organized, if we start with the gametophyte, so the gametophyte undergoes mitosis, mitotic divisions, and it leads to the formation of the male, reproductive structures or male gametes as we call them, which are called sperms, and eggs on the other hand, which are the female structures. There's a gametic fusion between the egg and the sperm, which leads to the formation of a diploid zygote. So if you look at this particular phase where the gametic fusion occurs, from this particular phase, the plants enter into the sporophytic, into the diploid stage. So this diploid zygote leads to several developmental stages leading to the formation of an embryo from which you have a diploid sporophyte. This is a progenitor for the diploid spore mother cells, which will in turn undergo meiosis, and from this point it leads to the formation of spores, which now enter the haploid cycle again, because they are ultimately going to give rise to the gametophytic stage. This cycle continues, and plants are so this particular character is so unanimously distributed among plants that you would find this everywhere. So this is a very, very important stage that they display both, a very important fact that they display both haploid as well as the diploid stage. Okay, so moving further, what are the other aspects? So the sporophyte is diploid and it leads to the formation of spores. The spores undergo mitotic division to produce the haploid gametophyte, which in turn yields the gametes. These gametes now undergo fertilization to produce the zygote. And this leads to the formation of the sporophyte, and this cycle goes on continuously. Okay? Now, what is this? So now we come to the pollen grain and the ovule. These are very, very important structures which are formed in the plants, without which the reproductive cycle does not get initiated. Now the pollen grain, which are formed in, these are the structures, if you have studied the structure of a flower, the flower consists of stamens and carpels, okay? The stamens, they in turn consist of the anther lobes which contain the pollen, okay? We normally refer to it as the pollen. So this is a structure which is formed as a result of development, okay? We'll come to that also. We are going to talk about microsporogenesis and microgametogenesis. So this is the structure of a pollen grain, which is typically comprised of an exine and an intine, which is an inner layer. It consists of a tube cell and a tube cell nucleus. As you can see, this is a very small nucleus. It consists of a larger nucleus, which is called the generative cell. Why is it so important to differentiate them as a tube cell and a generative cell? Both of these cells are destined to carry out specific functions in plants. So the generative cell, which you can see over here, it undergoes division to form two sperm cells. These sperm cells are now the structures which are going to fuse, or they are going to enter into the female embryo sac and fuse with the egg and lead to the formation of the zygote. So the zygote formation is something which is initiated by the pollen grain, but the sperms, they cannot, move and get or reach up to the egg cell. So that has to be helped by something. They need a channel, they need a portal for entry into the embryo sac. And that is what is actually done by your tube cell nucleus. So when the sperm cells form, one of them fertilizes with the egg and other joins with the polar nuclei, which leads to the formation of the endosperm. Okay. Then talking about the ovule. So the ovules are those structures which house the megasporangium, and the megasporangium in turn houses the megaspores. So the megasporangium, when they undergo meiotic divisions, they lead to the formation of 4 haploid megaspores. Now this is something which is very fantastic, which I always used to wonder, that there are 4 haploid megaspores. But in the end, what happens is one of them actually, leads to the formation of the final product. Okay. So all of the carpel is actually, the entire structure is actually diploid, but only for the megaspores, which you can see lined up one above the other, which is actually haploid. Okay. So this is ultimately going to undergo few more developmental stages and lead to the formation of an embryo sac, which we also call the female gametophyte. Okay. So let me go a little more further. And this is the structure of an ovule, which consists of, which shows the presence of the final embryo sac. This embryo sac. Now, there's one more thing which I would like to know from all of you. Looking at this picture, what do you feel? Is this embryo sac 7 cell or is it 8 cell? Some answers, please. I just hope nobody's sleeping. Yes, we are getting responses. 7 cells, 8 cells, 7 cells, 8 cells. Okay. Now, see, these are 3 antipodal cells. Okay. These are 2 polar nuclei. And these are 2 synergids. And in the center, you have an egg cell, right? If you look at the polar nuclei, they're actually separate entities, okay? But these separate haploid entities would fuse with each other. It is a prerequisite for fertilization. I hope all of you have actually read Fertilization. So the secondary nucleus has to be formed. So when the polar nuclei, they fuse with each other, they lead to the formation of a diploid secondary nucleus. And as we've stated in the previous slide, the sperm cells, one of the sperm cells would be actually fusing with these cells, okay? So it will not fuse individually with this polar nucleus and this polar nucleus. What it does is it will fuse with a diploid polar nucleus. I mean, the diploid secondary nucleus, okay? So this is a 7-cell embryo sac. Okay, if I have to say, the number of cells, then it is 7 cells, but the number of nuclei are 8. Okay, so a 7-cell embryo sac consisting of 8 nuclei. Okay, in this, one of the cells is the egg cell, which is surrounded by the synergid cells, which act as a supporting cell. And synergids are actually important. I don't know how many of you have actually read about the importance of synergid cells, but they have a very important role to play when the pollen tube actually tries to gain an entry into the embryo sac. I'll talk about it. I think in the next slide, and I'm going to show you a paper also, which is pertaining to the activity of the synergid cells. Nobody actually talks about synergids and antipodals, right? Because everybody gives importance to the secondary nucleus because it is going to form the nutritive tissue. And some other people, they just focus on the egg because it is going to form the zygote. But if these cells are not important really, why are they actually formed inside the embryo sac? You know, here there's a very important reference I have to make about the different types of development of embryos, okay? So different types of embryogeny, if you would have studied, there is a cleavage polyembryony, there's polyembryony, okay? So when these things happen, sometimes the sperm is not able to fuse with the egg cell. And in rarest of the rare cases, the sperm also fuses with the antipodals or the synergids. Synergids, the chances are very few because they have a fate of disintegrating just after the fertilization process takes place. but they are there to act as a stepping stone for the pollen tube to make an entry and release its sperm to fuse with the egg cell. Then the pollen tube enters the embryo sac by penetrating one of the synergids. So a penetrance into the synergid is extremely important to gain an entry into the egg cell. The central cell contains 2 or more polar nuclei, which would fuse with the second sperm nucleus. This is what we were talking about. And these are the 3 antipodal cells, okay? So what is the fate of these antipodal cells? Are they going to remain with the embryo or are they going to be disintegrated? So it has been studied and seen by the work of different researchers that these synergids, these antipodal cells, they tend to degenerate before embryonic development actually takes place, or sometimes they degenerate even during the embryonic development. So once the penetrance is over, once the zygote has formed, then they are no more needed, okay? So they get disintegrated or degenerated. This is a natural process. And then we come to a very interesting aspect of microsporogenesis. Till now, I have not gotten into the gene aspects, okay? So that will come very gradually. So microsporogenesis, as you can see, this is a cross-section, a transverse section through your anther, which is made up of, so here you can see 4 lobes, and in the center there's a vascular strand. So this is a process which defines how the development of your microspore mother cells takes place. What are the different structures which are formed, and what ultimately leads to the formation of pollen grains, right? So 4 somatic cell layers are usually formed during the process of microsporogenesis. The first layer, which is called the tapetum, I think you can just see my cursor around here. This is a kind of developing, a kind of nutritive tissue. Not a kind of, actually, it is a nutritive tissue which provides nutrition to the growing microspore mother cells, because these cells are ultimately going to form the pollen grains, okay? And this process actually is marked by the first stage, where it actually forms an archisporial stage where the cells are not actually differentiated. It proceeds further towards the development of a tapetum, the middle layer, okay? The middle layer are those layers which are just present below the, above the tapetum. Then the endothecium, which is a layer which is again lining and acting as a protective layer to protect the microspore mother cells which are present inside, and the epidermis, okay? The middle layers are actually many in number. So this will ensure that all of the spore mother cells should not undergo dehiscence, should not be moving out of the anthers before they undergo the development. So they need to be protected inside. So there is a lot of cushioning which is provided by the presence of the middle layers, okay? So except for the epidermis, which is a result of the L1 cells, the 3 inner somatic cell layers and the meiotic cells are usually generated from the L2, which is known as the archisporial cell. So here, the microspore mother cells undergo development till they lead to the formation of a pollen grain, which is usually referred to as a pollen tetrad. And when they attain maturity, you would find that these layers which we were just talking about, they gradually and slowly start disintegrating. So by the time your anther wall splits open and all these pollen grains are released outside, all of these structures are dissolved, okay? This is something which you've already studied, so let me go quickly ahead and talk about the genes. So this is a part which you all need to listen to very carefully. Now, most of the studies in plants have been done on the plant called Arabidopsis, as you all know. And that is because the system of Arabidopsis, the plant is very small because of its life cycle and because of the extensive, the ease with which the genes could be identified and studied. This has been selected as a very novel plant, or a model plant, I would say. So here, the product of 2 genes, the erecta gene, which is known as ER, and the erecta-like gene 1 and 2, so ERL1 and ERL2, These are two genes which have a leucine-rich repeat receptor-like protein kinase, which is known as the LRR-RLKs. And in the presence of these genes, it was found that anther lobe formation takes place properly. When Arabidopsis was subjected to mutations and the mutants were analyzed for the role of these particular genes, it was found that these genes, if they are repressed or if they are not expressed in the mutant species, the anther lobe formation does not take place. So these studies suggest that these are important genes which are required for anther lobe formation. The second gene, which is very important, is the excess microsporocytes 1, which is also known as EMS1. It is in combination with another gene, which is called the extrasporogenous cells, that is the EXS gene, which codes for, again, an LRR-RLK, which is required for the specification of the tapetum. Okay, so the previous gene cluster is required for managing anther lobe formation in plants, whereas in the second one, it is required for specifying the tapetal layer. So what happens is, again, when the mutant Arabidopsis species was studied, it was found that the spore mother cells fail to develop. They fail to undergo any division, and the anther does not dehisce. And this was because these genes, that is EMS1 and EXS, if they are not present, they will not allow specification of the tapetum. Why is it so important to specify the tapetum? The tapetum, as we already said, is that layer which is providing nutrition to the growing spore mother cells. Unless and until they are nourished, the development cannot proceed further. So this may lead either to the degeneration of the spore mother cells. This may lead to a non-division or an abnormal division of the spore mother cells, or it may also lead to no dehiscence of the anther. I mean, the anther does not undergo dehiscence, okay? Because if the spore mother cells are not mature, if the tapetum is not formed in the first place, there's no gene signal which is going to control the degeneration of the middle layers, of the tapetal layers, etc. Okay? So these set of genes are extremely important in specifying anther lobes and in specifying the tapetum. Okay? So highest level is found in the nascent tapetum, and there's another gene. So we now come to the next gene, which is called the dysfunctional tapetum 1 gene. This is known as DYT1 gene. Okay, this gene was found to be associated with a normal tapetal development and function. So this does not mean that in the absence of this gene, tapetum will not form. Okay, there is no stringent requirement for the presence of dysfunctional tapetum gene. This is a gene which would help your other genes to function and lead to the proper development, to the development of appropriate number of cells in the tapetum. And the next gene is the CAL-S5 gene, known as the callosynthase. Callosynthase is a gene which is responsible for exine formation. So when the pollen grains develop, they contain an outer exine and an inner intine, a structure which we have seen in probably slide number 4. So this structure is extremely important. Why? Not only because it is the outermost layer and it may be involved in protecting the innermost layers or something like that, but exine is a basis for taxonomical identification in some pollen grains. Pollen grains have a tendency to undergo ornamentations at the exine layer. So a lot of sculpturing and a lot of ornamentation takes place in some species of plants or in some plant pollen grains, and you could identify the pollen based on the ornamentation that they carry. So because of all these reasons, callosynthase gene is very, very important. And the second important function of this gene is to provide pollen viability. In the absence of this gene, definitely either the exine formation would be hampered, or the ornamentation process would be hampered, or the third thing which happens is your pollen grains may become non-viable or sterile. So these pollen which become sterile are of no use, okay? So here we've just understood all of the genes which are involved in this particular process. So with this, I move on to the first question of this session, right? So mutant Arabidopsis species lacks anther lobes and produces sterile pollen. So what would be the possible reason why it does so? Is it a mutation in AG? Here I would have to tell you what is AG. This AG stands for the A-gamous gene. A-gamous is a gene which is responsible for transition to flowering. I mean, it's a floral meristem identity gene. Second option is mutations in erecta and callosynthase. Third option is EX pistillata gene. Let me tell you here that pistillata is also a gene which is responsible for the floral meristem identity. With this, can we have the answers, please? B, 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 C. Okay. So most of you are answering it as B. Right. Yes, I have most of the answers stating that it is B. Right. So let us see what is the answer. So definitely it is erecta and callosynthase 5. Why is it so? Can it not be EXS? Because EXS is also one of the genes which are responsible for maintaining the tapetal identity. It cannot be, because see, when you read the questions while attempting it in your examination, be very careful to read what is asked. There are two parameters. One is the anther lobe, and the second is the pollen viability. Okay, so when we consider both these factors together, it has to be erecta and it has to be callosynthase 5, right? Okay, so very good for all of you, as most of you have, I mean, almost all of you have attempted the correct answer. Megasporogenesis. Let us very quickly get into megasporogenesis, because I have to focus a little more on the last two aspects, because that is what people have been concentrating in your CSIR-NET examinations. Okay, right. So megasporogenesis is basically a process where a diploid precursor cell, as you could see over here, or the megaspore mother cell, undergoes meiosis and it produces 4 haploid cells. This is something which we've seen in the previous slide also. What's so special about this process and why should it happen in the first place? So this is an example of a polygonum type of megasporogenesis. This has been observed. Arabidopsis is, of course, one of the examples, but there are other types also. The pattern or the type of cells which undergo degeneration is different, but the pattern of development, the way the cells divide, that is what makes them different, okay? So we have the monosporic, we have the bisporic, and we have the third type, so elisma and drusa and all of these types. So you have studied all these types. I'm not getting into the details of that, but what we are interested in is this particular megaspore. As you can see, the uppermost megaspore is actually very, very large in size. And what about the remaining 3? Why are they formed at all? So these are degenerated, and only this one actually develops into a mature female gametophyte. So what actually happens? So this is the process of megagametogenesis, right? Okay, we are still getting answers. I mean, the internet is slow probably. This is a process where you have to look into 3 important stages. The first one is the stage FG4. FG stands for female gametophyte. The FG5 and the stage FG6. So what happens here is that in the first case, you can see this is this cell, okay? We're talking about this cell. Now, when we proceed further, what happens to this cell? It undergoes division into 2, wherein a central vacuole is laid down. This division proceeds further till it reaches the first stage, which is called FG4. Okay, so in the stage called FG4, you have 2 nuclei which are separated by a central vacuole. Till this stage, we are talking about FG4 directly because till this stage, this vacuole is not very prominent, and it goes on increasing in its size as and when the embryo sac undergoes maturity. Okay, so this is separated by a central vacuole, and when it reaches the stage FG5, okay, that is this stage, the phragmoplasts, they tend to form, the phragmoplasts and the cell plates are laid down. This is the stage I'm talking about. So here you can see that between the sister cells and the non-sister cells, you would find that a plate is laid down. So this is a stage which marks a point where the cells are going to be differentiated properly. They are going to be separated, they're going to be segregated into antipodal cells on the upper side, right? The synergid cells on the lower side, a central egg cell, and the secondary nucleus. So what happens after this stage is one of this cell and one of this cell tends to cross the vacuum and reach towards the center, where it fuses and leads to the formation of the secondary nucleus that we have talked about earlier. Okay, so what happens is all of these cells are formed, and now you have a well-defined embryo sac. So what happens, yes, if the female gametophyte is not fertilized, the antipodal cells eventually tend to degenerate. So it is by nature that they're going to degenerate if there is no fertilization, or even if after the fertilization process gets over. Now, I think we are running a little short of time, so I need to really hurry up. So we now go to the next step, that is microgametogenesis. So actually this diagram depicts both the stages, that is microsporogenesis and microgametogenesis. So sporogenesis is something which we've already talked about, that is the formation of the spores. Now, yeah, I mean, I have talked about this part also. So development of microspore, that is the microspore mother cells, which we've seen in the structure of dehiscence of anther. So the development of this particular structure into microgametophyte, or the pollen grain which contains the sperm cells. So this is your flower, this is your anther, your microsporocyte, which undergoes division, leads to the formation of a pollen tetrad, of which each of the 4 microspores will now lead to the formation of a generative cell and a tube cell. Okay, and we have already stated what is the role of these individual cells. So one of them leads to the formation of the sperm cells, and the other one leads to the formation of the pollen tube. And both of these are important. One is important for fertilization, and the other is important to act as a channel through which the sperms can make an entry inside. Development of the male gametes. So this is already what we've talked about. So the vegetative cell usually grows and leads to the formation of the pollen tube, and the generative cell leads to the formation of 2 sperm cells. So this is what you could see in this particular picture. So this is your generative cell, this is your pollen tube, and this is the tube nucleus. Okay. I'll just go very quickly with this slide because we've been talking about this at length. And then comes the process of fertilization. A very, very important thing, okay? So here you can see that this is your ovary, these are your ovules, okay? This is the stigmatic disc, which is the surface of your carpel. So the pollen grain comes and sits on the stigmatic disc where it is recognized. So there's a lot of pollen stigma interaction which takes place. And there's a lot of incompatibility also, which is sometimes generated in plants. So pollen grains, when they sit on the stigmatic disc, they are recognized by specific receptors which are present on the stigmatic disc, which will recognize them and which will help the pollen grain to synthesize its pollen tube. So the tube nucleus now becomes active, synthesizes the pollen tube, releases the sperm, or it actually penetrates till it reaches this particular ovule where it encounters the female gametophyte. So when it actually enters into the female gametophyte, it catches hold of the synergid cells, okay? So one of the sperm is automatically directed to catch hold of the synergid and make an entry into the egg cell. And then the other one automatically goes and fuses with your polar nuclei. This is one more interesting feature of plant cells. Of course, in the human development also, this is a feature which is there. But in plant cells, what happens is that the process of fertilization itself takes care of formation of the nutritive tissue, which is a very, very specialized function. So all the growing embryos actually are nourished by this very nutritive tissue. And in some of the species of monocots, I mean specifically in monocots, you will find that this nutritive tissue, that is the endosperm, is still present in the plant. Actually, endosperm has to get exhausted by the time the embryo undergoes its development or completes its development. But in case of some of the monocots, it has been seen that it is still persistent. Okay, so yes. And now we come to another interesting part. This is another question, and these are not the questions from CSIR-NET. This is a question which has been put up to just check with whatever you've understood so far. So, sperm cell behavior during double fertilization in Arabidopsis can be stated as follows. Please read it carefully. Do not identify the correct answer. You have to identify the incorrect answer. So is it pollen tube bursting and discharging sperm cells? Is it sperm cells entering the female gametophyte via pollen tubes? Is it pollen tube entering embryo sac by penetrating one of the synergens? Or it is one sperm nucleus fusing with the egg cell and the other fusing with the central cell? Oh, A. Most of you replied as A. Okay. Can I know the reason why you are stating A? Does the pollen tube undergo bursting? Yes, can anybody answer that? Okay, one of them has also answered it. Yes. Any reasons why you think it is A? Okay, because all other options are correct. Okay. Oh, that's very obvious, right? Okay. Now, actually, I'll tell you what. Wait. The pollen tube enters the embryo sac by penetrating one of the synergids. Actually, I'll just correct myself over here. Here it has to be the antipodals actually. Okay. So the option should be C, but for C, it has to be antipodals instead of synergids. I just noticed that. So why is this answer incorrect? Incorrect because if I write antipodals, antipodals are not the ones which are going to be penetrated by the pollen tube. Pollen tube does not have any interaction with the antipodals. It rather tends to interact with the synergids. Okay. So as of now, the funniest part is all these answers are correct. Okay. In case of D, I would just like to clarify the D option. So what happens here is one sperm nucleus fuses with the egg cell and other fuses with the central cells. So the central cells are also known as the secondary nucleus, which is a result of fusion of polar nuclei. Okay, so that is why this answer is correct. So if you are confused at all, please get that clear. So it is also known as the central cell. Okay, right. And a justification for this is that your synergid cell is actually penetrated. And once it is penetrated by the pollen tube, it tends to undergo cell death. Its fate is to undergo cell death, just like antipodals. But it has a role to assist your pollen tube till the sperms actually reach the egg cell and actually fertilize it. Okay? So this is a paper. I hope it is visible to everyone. End of the presentation and end of the sessions, we are going to share all our presentations with all of you. So you would be getting some time to actually go through these presentations and understand them. So this is a paper which was published in Plant Cell. It's not a very recent paper, but this is a report which talks about the pollen tube, actually what is the mechanism that takes place and why does it actually disintegrate the synergid cells, okay? So this penetrated by the pollen tube undergoes cell death, either before its arrival or when the pollen tube contents are released inside. Okay, because the pollen tube contents are not simple cytoplasmic contents. They contain a lot of enzymes, they contain a lot of chemical substances, because there is an attraction of the stigmatic disc with the pollen grain. And this chemo attraction actually gives an insight into the fact that there has to be lots of other chemical reactions which are taking place in the cell. And because of that, the synergids are actually disintegrated. Okay, so with this, I move further towards a very, very important aspect. I think this was the third point that we had discussed. Yes, so this is about embryogenesis. Here we are going to talk about all of the development that takes place post-fertilization in plants. So this is a step which is aimed at three important aspects. The first important fact is that the development of a basic body plan takes place when the plants undergo embryogenesis. When I say a basic body plan, I am talking about the formation of independent structures, but all these structures are not patterned in a similar way. They have a definite way where how they are patterned, and they have a definite position on the plant body. So the first type of patterning that these cells undergo is radial patterning. Radial patterning is that which is responsible for forming the dermal, the ground, and the vascular tissue systems. Okay? Dermal tissues, when I say, I am talking about the epidermal tissues, that is the epidermis and the hypodermis. What is a ground tissue? I'm talking about the cortical zone. Okay, so the cortex, which may be parenchymatous completely, which may be sclerenchymatous, or which may be a blend of parenchyma, sclerenchyma, and collenchyma together. And vascular tissue systems refer to the stellar organization, the organization of the xylem and the phloem. The second type of patterning which takes place is axial patterning, and this takes care of the apical basal axis. So this is a patterning which takes care of the growth of shoots above ground and the growth of roots below ground. Also about how the nodes and internodes are formed, elongated, what is the nature of the shoot apical meristem and the root apical meristem. So all of this would be involved in, all of this would be a result of axial patterning in plants. The second feature of why embryogenesis should take place is to set aside meristematic tissues for post-embryonic elaboration of the body structure. So what is post-embryonic elaboration of the body structure? It means that the meristematic tissues, which are undifferentiated totally, they have to now get destined to develop into leaf, roots, flowers, shoots, etc. And the last important reason why this takes place is to establish an excessive food reserve for the germinating embryos. So how and why and what is the structure that I will talk about later? So when the embryo undergoes germination, it needs to have a food stock or a food reserve, because it has to undergo development for a long period of time. So it has to sustain. And for that, embryogenesis has to take care of synthesizing this accessible food reserve as well. Okay. Now, when this process, angiosperm embryogenesis, when it takes place, it starts with an asymmetric cell division. This asymmetric cell division gives rise to a terminal cell, which in turn gives rise to the embryo proper. On the other hand, it gives rise to a basal cell, which gives rise to the suspensor. So as we can make out, one is a terminal cell, which is situated to the upper end, and one is the basal cell, which is situated to the lower end. So naturally, when it undergoes development, and when I say that it is going to develop into an embryo proper, I'm going to talk about the shoot systems. And when I talk about the basal cell being responsible for the formation of suspensor, I'm naturally talking about something which is present or which is going to give rise to the below ground of the root meristems. Okay, so let us see what happens over here. This is a basic pattern of development. This is an example of a dicot embryogeny, but this is something which takes place in monocots also. When it takes place in monocot, I'll come to that fact later. Let me first talk about this. So this is your terminal cell and this is your basal cell. Basal cell, which is larger, okay? So these undergo divisions into a 4-cell stage. This 4-cell stage is extremely important because I'm going to emphasize on this particular stage in the next slide, where I'm going to talk about the Auxin gradients, okay? So this leads to the formation of a 2-cell embryo. The lower cell leads to the formation of a 2-cell suspensor. So this is followed by few subsequent divisions where an embryo proper is actually demarcated with a suspensor which comprises of vertically divided cells. Okay, so the lowermost portion divides into a suspensor, and by the time your embryogeny reaches a globular stage, so your embryo reaches a globular stage, what you find is that the embryo proper assumes a globular shape, and this assumes a foot-like shape. So that is why this particular stage is called a globular stage of embryo development. This now further differentiates into the structure which is called hypophysis. Development of hypophysis is very, very important because it is that part which is going to give rise to the embryonic roots. Understand this? This is important, okay? And the uppermost portion now assumes the shape of a heart. Why is it so that it assumes a chordate or a heart shape? This is because it is going to develop into 2 cotyledons, okay? So if it was one, it would be slightly just curved and twisted. But if it is 2 cotyledons, it is going to assume a very, very proper chordate shape. So here, your embryo is said to be heart-shaped or chordate shaped, okay? And after it reaches the torpedo stratification of the root meristem, this is your shoot meristem, and these 2 are your cotyledons, which by this particular stage become very, very prominent. And after a certain period of time, you would be able to see the actual cotyledonary leaves when it undergoes further development and when it germinates. Okay, now here I have put up a point which talks about particular genes. Investigations of suspensor mutants, that is suspensor 1, 2, and raspberry gene of Arabidopsis has provided genetic evidence which says that the suspensor actually has a capacity to develop into embryo-like structures. So then, if it has the capacity to develop into embryo-like structures, what is the fate of the suspensor? And what actually happens to these genes? Let us see this in the next slide. So this Sus1 gene, which has been actually found in Arabidopsis, has been renamed as DCL1 gene. Please remember, this is a very, very important point. I want all of you to note it somewhere, because when as a student I studied this particular gene, I studied it as the Sus1 gene. But now its name has changed to DCL1. Why is it DCL1? Because the name is derived from the term called dicer-like 1 gene. So its protein structure, which is predicted and studied, it resembles the dicer gene in Drosophila melanogaster, and it is also similar to the DCR1 gene, which was studied in Synorebbatis elegans. Okay, so because of its very close resemblance with these 2 genes, it is now known as the DCL1, which is a hybrid name, which is derived from the dicer as well as the DCR1. Okay, so these mutants affect ovule development and transition from vegetative to reproductive development. So when the Sus1 gene is present, it will actually take care of ovule development. And when the plant transits from vegetative to reproductive development, then it actually is helped by the Sus1 gene.
Yes, Dr. Kota, one question is there from Twinkle Shah.
Okay.
Yeah, the question is, as plants do not contain microtubules but phragmoplast, what makes the terminal and the basal cells to divide in horizontal and vertical planes respectively?
Okay, got it. Yes, so what happens here is that first of all, if the question is pertaining only to phragmoplast, so let us concentrate on phragmoplast, okay? So phragmoplast is actually, see, there are lots of features which actually determine this kind of, your question is related to what? The arrangement of terminal and basal cells, right? Okay. Are you talking about the terminal cell where we started with and the basal cell, the single cell structure? Because when the single cell structure is actually dividing into two and those two into four, that is all totally under genetic control. There are lots of genes which actually control this particular factor. And in response to your question, what I feel is this phragmoplast is actually responsible for, along with the genes. So this is a coordinated activity where these two together will determine as to what will be the patterning system. Okay, I have not gone into the details of the genetics behind the pattern of patterning, which determines where the polar nuclei will migrate and where they will reach and all that. So, but then yes, I think I can get back to you if you write to me specifically in an email, and I will also share some papers which will give you a deeper insight into that particular part. But yes, it is a phragmoplast along with certain specific genes which are going to actually determine whether it is going to be cut in a vertical or a horizontal plane. Okay, right. So with that, I'll move ahead with the radial and axial pattern. Radial patterning is something which emerges at the globular stage. So as you can see, this is the globular stage of the embryo. And globular stage, because globular stage is that where the complete identification of a cotyledon has not yet been reached. Okay. So this is something where only the specification into dermal tissue, ground tissue, and vascular tissue is initiated. So when radial patterning happens, the outermost layer, that is the protoderm layer, is the one which is going to be responsible for the formation of epidermis. So what would be the nature of epidermis, and why is epidermis so important? Because as we all have studied anatomy, we have seen that epidermis is just a single layered structure. But if you have studied plants in details, there are certain plants which contain hair, which contain epidermal hairs. So the patterning and the differentiation of these epidermal hairs is also important. So that is something which is coming as a result of radial patterning. So this takes care of the formation of epidermal layer. Just a second. Yeah, fine. And then this is followed by the ground tissue. So the ground tissue is just as I said, whether the ground tissue is going to be parenchymatous, sclerenchymatous, or where there is a deposition of suberin sometimes in some plants. There are raphides in some plants, sclerids in some plants. Okay, so whether there is any kind of deposition which is to take place. So all these things, but of course, the deposition is something which is going to be taken care of by some other genes which specify the synthesis of these chemical substances or these structures. But during patterning, actually what happens here is that the epidermis is distinguished from the ground layer, and the vascular system also is distinguished, and they take their respective positions on the plant. So epidermis, which forms the outermost layer, followed by the ground tissue, which is in turn followed by the vascular system, which has to be situated centrally into the plant. So this kind of a patterning is what is defined by the radial patterning. Then axial patterning is something which is actually determined at the heart stage or the chordate stage, which is a step ahead of the previous stage. So what happens here is that the globular shape of the embryo now is lost in the form of cotyledons. So when the cotyledons form, the globular embryo transits into the chordate shape. So dicots usually have two cotyledons, okay? And in monocots, there's only a single cotyledon. But what is important to understand is not about the cotyledons, but that in this particular stage, when axial patterning takes place, it gives a clear-cut identification of two cotyledons which are present in dicots. On the upper end, as you can see in the picture over here, the shoot meristem is specified, and on the lower end, the root meristem is specified with the procambium in the center. Okay, so shoot meristems are specified at this point because lots of activities has to take place at the shoot meristem, which not only involves leaf formation, but it also involves what would be the length of the stem, what would be the nature of the stem, So all that is going to be specified at a later stage. But for the shoot meristem to become determined or destined to actually lead to the formation of different individual structures, it is extremely important that the patterning takes place at an early stage. Root meristems are also important because they are going to specify the formation of, for example, the root cap cells or the columella or the other structures which are organized. Okay, so basically axial patterning would give the plant a clear-cut identification into where the cotyledons are going to be present, where the shoot meristem and root meristem is positioned, and where the brocamen is positioned. Okay, yes. So this is a very, very important slide. I want you all to really focus and understand this. This is a slide which basically talks about the, just a second. Okay. Yeah, students, I would just suggest that I'm not able to see the questions in between. So if at all you have any questions, you can directly post it to me.
Yeah, ma'am. One question is there.
Yeah, I'm just reading it. Yeah. So if root meristems are situated in the embryo, then what does the suspense part include? Okay. See, this is where the root meristem is actually situated, right? So this was developing from the suspensor, which leads to the formation of hypophysis. Once the hypophysis is formed, it is after that that the root meristems are specified. Okay? So I hope that satisfies your answer. And with that, I quickly move over to the actual topic on Auxin. Yeah, students, just pardon me, I'm not able to read it anymore. I hope you have got the answer. Yes. So I'll now go towards Auxin development, okay? Auxin development, this is a very important topic because this has been asked in 2 of the CSIR-NET examinations, and that is why I want you to understand it actually. So this is a feature of auxins. Auxins, as we all know, are plant growth hormones, which are responsible for apical dominance in plants. There are so many other characters associated with auxin development and auxin expression. But what is important is the role of these PIN and the PID genes, okay? So it has been studied again in Arabidopsis that very early in the stages of embryogenesis, there's a family of these pin form proteins which are known as the pin proteins. Okay, so these proteins act as auxin efflux carriers. What do you mean by auxin efflux carriers? It means that they are responsible for creating effluxes of auxins in and across the plants. Okay. So when I say plants, I'm actually talking about the organized structures. But at this stage, I'm just simply talking about the different developmental stages. Okay. So in an embryo, these pin-form proteins, they are actually asymmetrically distributed. They're present in whatever amounts, but they are asymmetrically distributed. Now, there's another protein which is called pinoid protein, which is known as the PID protein. Okay, so this pinoid protein, known as the PID protein kinase, it actually aids in asymmetric localization of the pin proteins. So, when the plant, when the embryogeny occurs and it reaches the 4-cell stage, the auxin tends to move at the 4-cell stage, as you can see in the picture over here. So this is the 4-cell stage. I was talking about this in the previous slide, right? If you remember. So the basal and the terminal cell both have undergone division into a 4-cell stage. So here, your auxin tends to move apically. But by the time your embryogenesis proceeds further and reaches to the globular stage, the efflux carriers which are present, it tends to actually direct the flow of this particular auxin towards the hypophysis, which are the cells which are situated below. Not in the region of the embryo, but rather away from that direction. Okay? So here, when the auxin effluxes move towards the hypophysis, it is going to lead to the developmental aspects. I mean, it is going to have an impact on the developments of the root meristem. Now, looking at this picture, so here This red arrow indicates the auxin transport, which is actually done by the pin family. Okay, these are the cells which are actually accumulating auxin, and these are the cells which are actually producing auxin. So you can see that they're produced elsewhere, but because of the activity of the pin and the PID proteins, their effluxes which are generated elsewhere are carried to other parts of the plant. Now this first picture depicts a wild type Arabidopsis. Here you can see that the concentration of the spin-1 auxin efflux carrier is present in this particular region of the embryo. Okay, so yes, so disruption of the auxin morphogenetic gradient, either through mutation or overexpression of PID, allows this auxin to accumulate more apically, and because of which your root development gets hampered or it becomes abnormal. So what happens? So in this picture, you can see there is a lot of localization of auxin in this region. Okay, but when PID overexpresses, it tends to have an impact on the activity of your pin effluxes. Okay, pin-1 effluxes, sorry. So this leads to more of, yeah. So here, yeah. One second, yes. So in case of the first picture, concentration of pin-1 auxin efflux carriers moves towards this direction. So here, the expression of your auxin is more towards the basal end, okay? Whereas here, when PID overexpresses, it actually tends to block your pin-1 or move its effluxes across the cell, because of which what happens is most of the auxin which is synthesized or which is expressed, it gets concentrated in this particular region. So the ultimate product of this mutant is that auxin-induced gene expression is more in this region rather than the shoot region where it should have actually been. So if you study any of the mutants which are showing an auxin-induced gene expression more in the embryonic region compared to that in the root meristem region, then that is definitely an expression, an overexpression of the PID proteins. There's a lot more actually to be discussed about the PID proteins and PIN proteins. I'm just simply focusing myself to what they actually do to the cell, okay? In the end, I have attached some reference papers and I would like all of you to really go through these papers from where I have even taken these references. They will actually help you to gain more insight into this topic, right? And this is the reference that I was just talking about. So this is again a paper which was published in the journal called Plants. This is a very recent paper of 2019 where the diverse roles of auxin in regulating leaf development is given. I would suggest all of you to kindly go through these papers. Please don't take it just as a suggestion. Please go through these papers and please try to solve your papers based on them. So very quickly, let us get to the next question, which is what is the factor responsible for early embryonic access during early developmental pathway? I think this should be very simple. This was a question which had appeared in the CSIR-NET exam of December 2008. And it's still being asked, okay? Don't think it's a very old paper, so it will not be asked. Yes? A. Okay, I think the answer is very, pretty simple. See, orientation of embryo sac. How is it going to actually determine embryonic axis? Plane of cell division. Plane of cell division up to some extent, yes, but it is not ultimately going to determine anything related to the axis. Morphogens. Morphogens are responsible for morphogenetic patterning and differentiation, okay? So obviously, as you all have answered, I cannot see your names right now, but all those who have answered have unanimously answered it as auxin gradient. So I think that particular aspect is clear to everyone. Can we have the next question, please? This is also simple. Yes? Okay, I'm getting some answers again. I'm still getting A. I hope you're not answering the previous question, right? Okay. Okay. So let me not put you into any more suspense. The answer is actually simple because these are responsible for what? Auxin effluxes and establishment of auxin gradients. So well done, all of you. 2011A. Oh, you have put the year of the paper? Okay, fine, no problem. So with this, now we get into a very serious topic. I just hope I can do justice to it because there's very less time remaining. And please listen to this topic extremely and very, very carefully because this is a very favorite question of the paper setters for CSIR-NET. Transition from vegetative stage. When plants actually undergo transition from vegetative to the reproductive stage, lots and lots of changes take place, and they are mostly under the control of several important gene products. Okay. The first important gene product. Now, I'm not getting into the background of anything. I'll just simply focus on what I want to talk about. Okay. I hope you will be able to understand the background. If somebody has any issues in understanding the basics, you can get back to me later, right? The first important gene is called leafy. You'll have a reference of this leafy hereafter, wherever we talk about floral meristems, okay? So this is a gene which is very importantly responsible for the plant to transit from vegetative to reproductive stage. This has an additional job of actually specifying compound leaf development. Everybody knows that plants have simple as well as compound leaves. So compound leaf development is where the leaf actually gets subdivided into smaller leaflets. This is also a genetic pattern in plants. Sorry, morphogenetic pattern in plants, okay? So leafy is a gene which is responsible for helping the plants to transit from vegetative to reproductive. So I think so far we all always felt that the plant is naturally going to undergo development, right? Gene hai ya nahi hai. Agar leaf form hua hai, तो then naturally the leaf is going to transit to flower. But that is not the case. If at all there was a mutation or underexpression of the leafy gene, there will be no transition and the plants are maintained in a vegetative state. This actually happens in plants where there is something called a kind of dormancy. Okay, dormancy is a term which we normally use to refer to the buds or to the seeds. But here what happens is the plants are not required to undergo flowering if the conditions are not appropriate. So in that condition, leafy will remain suppressed. Okay, this is a gene which was unanimously identified in Arabidopsis as well as in Snapdragons. But in Snapdragons, we actually call them floricola. Snapdragon is antirrhinum, and it has homologs in other angiosperms, which may be named differently. So there are actually papers which are pertaining to maize. There are papers which are pertaining to other plants also. So there, the gene partners would be different. So in Arabidopsis floricola and in Arabidopsis leafy and in Snapdragons floricola. The P homolog is called unifoliatum. Okay, so the P homolog actually has a negative activity here. Here it actually reduces the leaf, compound leaf to a simple. Okay. Now, establishment of meristems. This is a prerequisite for any transition from vegetative to the reproductive stage. Okay. So meristematic cells, they are the cells which behave like stem cells in animals. These are the cells which are totally undifferentiated, but they have a tremendous capability to undergo differentiation. In case of plants, they undergo differentiation at many different levels and into many different cell types. And that is why we find such a lot of diversity when we talk about plants, okay? So the root meristem is partially derived from the hypophysis, as we've already seen in case of embryogeny. Older parts are derived from the embryo proper, which is situated above. Apical meristems occur at the growing shoot and root tips. Root apical meristems, they tend to produce an additional structure which is called the root cap. And the shoot apical meristem tends to produce stem, leaf, as well as reproductive structures. Okay? So root apical meristems, I would say, has very few amount of differentiation going on, very less amount of differentiation going on, where it actually tends to form the columella, the root cap, the quiescent center, and very few structures which are associated only with the development of roots. At the same time, I would not fail to talk about the role of root apical meristems in actually determining what would be the type of root. So in plants, you have the taproot, the adventitious roots, and the fibrous roots, okay? So all of that is going to be derived from the root apical meristems. So yes, it is also a very important type of meristem found in plants. So talking about the root apical meristem, so here, as you can see, this leads to the formation of lateral roots. So from the primary root, the lateral roots are established. A very important feature of the root apical meristem is actually the presence of a particular region. Okay, so let me first concentrate on what it contains. So the apicalmost region consists of a group of cells which forms the root cap. The root cap is basically a protective layer which is present only up to a certain period of time, after which it degenerates. So actually it has to be present to protect the root from any kind of mechanical stress, injury, or preventing it from withering off because of scarcity of water, etc. So after its role is done, that gives away. So that withers away. So immediately after that, you find a region of cells which actually forms your apical meristem. Okay, so root cap cells are fully matured cells which either develop from the dermatocalyptrogen. So the layer which actually develops the root cap cells in dicot plants is called dermatocalyptrogen, and in monopod cells it is formed from the calyptrogen. So usually it leads to the formation of it leads to the differentiation of other tissues, because plant roots also have a stellar system. So it has a procambium, it is a ground tissue, protoderm, pericycle, root hairs, lateral roots, etc. And this is a structure that I was talking about. So there is a very typical group of cells which is just present, next to the apical meristem, and the cortical endodermal stem cells form a ring of cells which surround the quiescent center. So a region of cells which is actually not going to divide any further is called quiescent center. There are lots of theories which are pertaining to the development of the root apical meristem, one of which is called the quiescent center theory, which talks about this particular group of cells. Okay, so yes, so it does not divide after embryogenesis takes place, and it remains as it is in the plant. So we can say that it is a zone which demarcates or which separates your root cap cells from the apical meristematic cells. Okay, so the cortical endodermal stem cells, which are found in the region which is surrounding this quiescent center, it tends to represent the cortical and the endodermal layers, which means the layer which is present below the epidermis, and endodermis which surrounds the pericycle, which is very much a part of the stellar organization. Okay, and the columnar cells, columnar stem cells are those which are situated immediately above the central cells. Okay, so this is a region, and they divide anticlinally and periclinally, and they actually divide. So this is actually the region, and it generates a sector of the root cap which is called the columella. Okay, this is all very common. I think I'll just go very quickly with these things. Yes, the shoot apical meristem. A very important thing that whenever angiosperm shoot apical meristems are organized, they are organized into 3 layers: the L1, L2, and L3. Okay, and L1, L2, and L3 actually determines one layer specifying your dermal region, one layer specifying your cortical region, and one layer specifying the vascular region or the vascular elements. This is important. Mutations in Arabidopsis clevata gene. There's a gene called clevata, which was also studied in Arabidopsis. So it was found that when there is a mutation in this gene, it leads to increased meristem size, and it also leads to the production of extra organs, which means that the CLV1, CLV2, and CLV3, which were the specific genes which were studied, it limits the number of undifferentiated stem cells in both vegetative and floral meristem. So what happens when the number of undifferentiated cells is limited? So it means that it goes, or it takes the cell, the plant body, more to a state where it is going to become determined or committed. Another set of genes are the VUS, the POL, and the STM. So, sorry, these are the proteins. The wasp is from the WUSCHEL gene. So these are those proteins which act to keep the meristems in an undifferentiated state. So can you say that this is a gene which is actually limiting, and this is a gene which is actually acting antagonistically and keeping it in an undifferentiated state? So one is actually required to be expressed when the meristematic cells have to undergo commitment to produce a particular organ of the plant. And the other one is required when organ development has to wait for quite some time. So when a plant is developing into compound leaves and a more intricate type of floral arrangement or a type of phyllotaxy, it needs a lot of time, okay? So the differentiation, though it takes place, it has to take place at a slower pace. And plus, it depends upon the developmental stage also. If it is in the vegetative stage, you need more and more number of meristematic cells because still it has to undergo reproduction. Root development. Root development, when it was extensively studied, it was found that there are two gene products which are very important over here. The scarecrow gene, which is known as SCR, and the short root gene, which is known as SHR. Okay? So SCR gene actually tends to be important for asymmetrical cell division. And this asymmetrical cell division actually is, I cannot say phenotypically, but anatomically it is seen in the form of a smaller endodermal cell, whereas there is a larger cortex cell. Okay? So endodermal cell is not going to be very thick, rigid, and very large, but usually it is small, but here it is smaller. SHR is a gene which is responsible for specification of the endodermal cell, which means even if SCR is present, and if SHR by any means is not present or not expressed, it means that the specification of the endodermal cell will not take place. So it is a combined and a combined activity of the SCR and SHR, which will determine what would be the nature of these two regions, that is endodermis. So endodermis in turn will also determine the nature of the pericycle, okay, and the vascular elements situated inside. But of course, these genes have nothing to do with the vascular elements, right? And shoot development is specified by the WUSCHEL gene, which actually specifies the identity of the stem. I'm simply now going to focus only on the genes, okay? So the genes which are responsible for shoot apical meristem in Arabidopsis have been studied as the CUC1 and 2, which is actually referring to the cup-shaped cotyledon. It is actually referring to the shape of the cotyledon on the basis of which, because it controls this particular morphological character, it is known as the CUC gene, okay? Then the shoot meristem gene known as STM and the WUSCHEL gene. Very, very important, okay? And phyllotaxy is another important aspect. I have no time to talk about phyllotaxy, but yes, it is a very, very important aspect of plant development because each plant will have a different arrangement of leaves. And it is also something which is going to be controlled, which would be under proper control of genes. So the PIN gene, I'll just simply, talk about it, the overview of the adaxial and abaxial leaf surface formation. As we all know, adaxial and abaxial is important because it is responsible for the localization of stomata, distribution and localization of stomata, and also the type of stomata that the plant has. So the PIN gene, remember the PIN gene where we were talking about Auxin effluxus? So the PIN gene is one of the genes which is related to phyllotaxy. And this is its localization. The localization of Auxin, which is actually caused by PIN genes, is a very important aspect in determining whether the adaxials, what would be the nature of the adaxial and abaxial surface. So PIN efflux carrier concentrates Auxin into the local maxima in the epidermis. Okay, so when it concentrates maximum Auxin in the epidermal region, it positions incipient, which position the incipient leaf on the floral primordium. I'll simply get into the diagram over here. Okay, the diagram comes next. Okay, can we just quickly solve this? This is also very easy, okay? Or I think I'll simply give you the answer. Is that fine? so that you can get an insight into the ABC model, which is important. Okay, so here the answer is clavata. Please don't say leafy, okay? Leafy is responsible for the floral meristem gene identity. But here the question is gene responsible for shoot meristem formation. We are not talking about the meristem identity, floral meristem identity. We are talking about shoot meristem identity. So the correct answer is clavata. And leaf meristem, leaf development is specified by the formation of leaf axis, okay? And then morphogenesis, which takes place giving rise to different types of leaf shapes and also the different types of phytotaxy, okay? And the unique shape of leaves is actually a resultant of cell division and cell expansion. So steps in development, yes. Okay, there's a lot of things which are to be talked over here. So I'll simply talk about the two important aspects, that is dorsiventral and proximodistal development. So here you have the abaxial-adaxial arrangement. As I said, this is important for light absorption and for many other features, stomatal openings, stomatal closing, okay? So here I'm just skipping. So L1 layer, when the leaf develops, L1 layer differentiates into epidermis. The other one differentiates into the photosynthetic mesophyll cells, which form the central core of the leaf, and L3 layer differentiates, as usual, into the bundle sheath cells. Okay, bundle sheath cells are the cells which surround the vascular bundles. Yes, this is what I wanted to talk about. So the fabulosa gene. Okay, this is a very important aspect for you all to understand. So fabulosa and favulota are a combination of two genes which are responsible for an adaxial signal. There's another gene called the Kennedy gene or the CAN gene. Okay, this is a gene which initiates abaxial differentiation in Arabidopsis. This CAN is actually transiently expressed on the abaxial side of the plant. So what is adaxial? Adaxial is the upper one and the abaxial is the lower side of the leaf. Okay. So when there is an overexpression, when there is a proper expression of the CAN gene, the abaxial surface is specified. So this is expressed not only on the abaxial side of the leaf, but rather the cotyledons, the leaves, and the initiating floral organ primordia also. There's another gene called YABI, and YABI is a helper of the CAN gene, right? So this helps it in actually specifying the abaxial fate. And then CAN gene expression is restricted to the abaxial side by PHB and PHV. So it has been seen that when PHB, and CAN, they act together, they act in a manner where they tend to have an antagonistic expression on each other, antagonistic activity on each other. Okay, so CAN actually is restricted to abaxial side by the PHB and PHV. On the other hand, the CAN actually restricts PHB and PHV expression to the opposite side. Okay. So once again, my slide is not moving. And this is what the picture talks about. So in the first picture, you can see this is the expression of PHB and PHV over here. So microRNAs, which are expressed on the abaxial side, it tends to bind to the PHB and PHV RNA. Okay, so here the adaxial surface is specified. This is a second picture which shows the expression of CAN, which is helped in by YABI genes. Okay, so which initiates the transcription of other abaxial genes. So the abaxial part is specified. So here in this picture, the PHB and PHV in the third one, as you can see, they are antagonistically acting upon each other. So PHB and PHV block CAN adaxial activity. So any activity of CAN on the adaxial side and any activity of PHB and PHV on the abaxial side is going to be blocked when they act together or when they act antagonistically with each other. Okay. Yes. And this is the last part, which I have to be really fast with. So the gene leafy, as we already discussed, is responsible for vegetative to reproductive development, and floricola is the gene which is for studied in Snapdragon. So these are the changes that take place, okay, when the transition takes place. So juvenile vegetative growth to adult vegetative growth, inflorescence growth and flower formation. And phase change genes actually help in this transition. Floral meristem identity genes help in transition from vegetative to inflorescence. Floral organ identity genes help in flower formation. So this is a later stage, okay? So leafy is a gene which is responsible for floral meristem identity. Now, when leafy interacts with another gene product, which is known as apetala-1, it promotes the transition to floral meristem. Normally, leafy is expressed in very low levels because it is to actually go on decreasing its content so that the plant transits from vegetative to reproductive state. Okay, so that is why we are saying that it is expressed at low levels in the vegetative tissues, and its expression is strongly upregulated when there is a floral inductive signal. Floral inductive signal may be in the form of hormone called florigen. It may be in the form of vernalization, which refers to a process where there is a low temperature which favors flowering, okay, or any other factor which actually favors flowering. So plant mutant for leafy do produce axillary meristems which possess floral identity because there is another gene called apetala-1. So normally leafy plus apetala-1 both will be, determining when the plant has to transit to the floral, the vegetative to the reproductive phase. Okay. So loss of functional mutants in AP1 shows that there is a partial conversion of these floral meristems into a more inflorescence-like activity. So that is actually a transition stage. So when there is a loss of AP1, then this change into a flower does not take place and it is halted for a particular period of time. And where there are leafy and AP1 double mutants, there is no flower formation at all. So this indicates that both of these are absolutely responsible for specifying the floral meristems. Agamis encodes a factor which is called the Matchbox transcription factor. If you remember the homeotic genes that I was talking about in the very first slide, there I made a reference of these Matchbox transcription factor. So these promote the determinate development of the floral meristem by limiting stem cell proliferation. Okay. There's another gene which is Wuschel gene. I think this is a gene which is there in animal development also. This is a homeodomain containing transcription factor which is expressed at the organizing center of the shoot apical meristem. So when Wuschel gene is present, it maintains a lot of stem cell identity because this is the one which specifies more of meristems. Okay. So Wusch and leafy, they tend to bind to the adjacent sites where Agamis gene is present and it promotes the upregulation. And activation of AG tends to have a negative feedback. So this results in a down-regulated stem proliferation and more promotion of determinacy, which means that stem cell proliferation, when it is down-regulated, obviously there will be less amount of or loss of meristematic activity, or there will be more amount of meristems which are going to become determined. Okay. So I'm not asking you to give me the answer right now. Let me just get to the question. So which of the following statements is true about leafy? Okay. So it is involved in floral meristem identity, leaf expansion, meristem identity, or far red light mediated seedling growth. So it is floral meristem identity. Okay. This is a question of CSIR-NET 2015. And then we come to juvenility, which is a very important phenomenon, which is actually seen in some of the woody plants and some of the plants which undergo secondary growth. These plants actually have to wait for a phase which is called a juvenile phase before they actually produce a reproductive structure. So in case of these plants, even if there are all the factors which are conducive for flower development, they still compulsorily undergo a juvenile phase. This juvenility is maintained by a certain specific set of genes, which are known as the early phase change genes. So it was found that in case of maize, the EPC gene known as early phase change gene is responsible for maintaining juvenility. This may be for many reasons, that it is not yet ready to undergo flower development, or if it undergoes flower development very early, the seeds which are produced may not be viable, they may be sterile. So many reasons are there for juvenility, okay? And in Arabidopsis, the juvenility gene is called the hasty gene or the HST gene. So as I said, there are many structural analogs of these genes when it comes to different plant species. So there's a small organic flowering molecule known as florigen, which activates the process. Leafy actually causes the meristems to get into the mode of development of flowers. Okay. mRNA or protein product. This is important. I'll just put up all the points together and then I'll talk about it. Yes. There's a protein product of the flowering locus, which is known as FT. This FT is a very positive regulator of flowering, which means that when FT is expressed, flowering actually takes place. Upon reaching in meristem, FT actually interacts with another transcription factor, which is known as FD. Okay, so FD stands for the flowering locus T. So there's a strong interaction between this FT and the FD. So there are also reports which suggest that this FT protein is itself acting as a florigen. What is florigen? Florigen is a flowering hormone. So when FT is produced, it gets transported to the shoot apical meristem. And when it reaches upon the shoot apical meristem, it encounters and interacts with the FD gene. Okay. And then there's a third gene, which is called Constance gene, which is known as CO. CO is a gene which responds. I have particularly taken a reference of CO because it is also there in some of the upcoming questions. Okay. So it is a gene which responds to daylight. It promotes flowering under long day conditions. And it is also responsible for activating your FT. So indirectly, just as FD activity is related to FD, it is also related to Constance genes or CO gene. So among which of the following, which of the following gene products migrates from leaves to shoot? Meristems. Okay. So is it the flowering locus T? It is the flowering locus D. What is it? So the answer here is leafy. Again, okay. T, D, apetala alone is not responsible for anything. It is always leafy, which is responsible for, causing this migration. Because as we said, it has an activity in the shoot apical meristem, converting it into the floral meristem identity. And this is, I am relieved now because we have reached the ABC model, which is the most important topic. So flowering is basically something which is a characteristic feature of three important genes. There's actually a fourth gene. I'll talk about it. Class A, Class B, and Class G. And when the Class A, B, and C genes were studied in Arabidopsis, they were found the Class A genes are the AP2 genes, apetala 2. Class B genes are AP3, that is apetala 3, and PI, which stands for pistillator. Class C is AG, which stands for A gamma G. Okay. So when Class A gene is specified, it leads to the formation of sepals. Class A and Class B together specifies the formation of petals. Class B and C actually specifies the formation of androphore, and Class C alone specifies the formation of carpels, which means that when there is an expression of Class A and B, the plant will form petals. What will happen if there is Class A and B alone? I mean, Class A and B. So there will be an expression of sepals and petals. Imagine a situation where there was A and C. So when there is A and C, there will be the formation of sepals, there will be the formation of carpels. As B is absent, there will be no petals, and as C is absent, there will be no stamens. So this is all, coming in from a very important study on the MADS genes, which is a highly conserved class of homeotic genes. The name MADS actually is derived from a combination of these four, that is MCM1 from yeast, A-gammas from Arabidopsis, deficients from Snapdragon, and SRF from the Homo sapiens. Okay, so as stated earlier, these are the genes which were specified in the previous slide. So class A and specifies sepal development. Now this is something which I've already talked about, so I'll not repeat it. A fourth class of floral identity genes, this is what I was saying, is called sepalata, which was identified in the absence of which flowers become complete violet sepals. Okay, so sepalata is actually a gene which was identified later, but still there are lots of studies which have to be done for this particular gene to be proven as the class D gene completely, okay? There are reports which suggest that it can act as the class D, but still lots of more studies are required for this, okay? So this is what. And the last aspect is just a comparison of what happens in Antirrhinum, okay? So this is Antirrhinum major Snapdragon flower, in which the flowers are bilaterally symmetrical. This is important. The flowers are zygomorphic, so there has to be some additional genes which actually specify this kind of asymmetry in the flower. So when there were studies which were done on Antirrhinum, I'm sorry, I skipped the slide. Yes. So it was also seen that this is not the only factor of concern. But there's another factor, another point of concern, which says that there are 4 stamens, out of which 1 is degenerated. So these flowers produce 4 mature stamens per flower. And the 5th one, which is actually produced during development, it gets aborted at a later stage. Okay? And here, the petals show a lot of asymmetry, wherein you can see that the AD and these are 2 AD, these are 2 Ls, and this is the one which has a bilateral symmetry. The central one has an exact bilateral symmetry, but the other petals are totally asymmetrical. Okay? So there has to be, definitely there has to be different genes which specify this character. So floricola, as we already said, is a gene responsible for floral meristem. This is promoted by squamosa and apetala 1. Apetala, which is analogous to Arabidopsis. The Wuschel gene is one which is responsible for clevata. Clevata, as we already studied, it is also a gene which is synthesized in case of Arabidopsis. Okay? So this is a gene which actually tends to control the number of stem cells that are produced. And this particular regulation is important for terminating meristem growth so that the plant actually gets into the, it moves out of the vegetative system and gets into the floral meristem identity. And closely related to more genes called cycloidea and dichotoma. They are actually, this is important, they are responsible for development of zygomorphic flowers. Okay, and whatever is the difference in the nature of the adaxial and the adaxial identity of the floral organs, that is the asymmetry of petals, as well as the adaxial stamen abortion. All of these are under the control of the genes called cycloidea and dichotomy. Okay, so they are known as the CYC and the ditch gene, respectively. So ditch actually confers distinctions between halves of dorsal petal, making each petal more and more asymmetric. And what is the role of CYC? So CYC results in the lower 3 petals developing as mirror image duplications of the lateral half of the dorsal petals. Okay, so here we are talking about this and this. Okay, being the mirror images of each other, and this also actually, if you look at the petals, they are mirror images of each other. And the dorsal and lateral lobes are individually asymmetric, while the ventral petal has a bilateral symmetry. And this is a reference of this particular aspect. Please refer to this paper, which is also a paper which is very recent, published in 2019. This is the only paper where you can actually find all of this information pertaining to all of the genes responsible for antirrhinum development. Okay, and this is the last question, and I think you all will be able to answer this, right? Can I, because we have ended with the session, can I please have all of you answering this question? Are you all there? Still listening to me or not? Because I have not got any responses so far. Yes, okay. So shall I give you the answer? I don't think anybody, oh, okay, answers are coming. Okay. So you say that it is apetala, 1 leafy and suppressor of constans 1. Okay. Anyone else? Actually, I think since the question is long and all the statements are different, you need to actually work it out on paper. Like what is the role of epitella, what is the role of leafy, what is the role of constants, and what is the role of suppressor of constants, right? Okay, so can I go ahead and answer the question? I hope so, because we are running short of time also. Yes. So the answer actually here is two major type of genes regulate floral development, meristem identity and floral identity. Isn't the statement true? Actually, I forgot to put the statement, which of the above combination is correct? That is the question, okay. I thought I forgot to put that statement. Second one is about epitella, leafy, and suppressor of constants. Yes, it is true. And the third one says that these are the genes which were discovered as floral homeotic nutrients. What is wrong with D? What is wrong with D? D is wrong because it is not FAD, okay? It's the max box genes. That is what makes it so wrong, okay? Right. So having said all this, I know you all had a very exhaustive session. I would like to tell you here that the book by Gilbert on developmental biology, the 2010 edition of this book actually contains a separate article on plant development. And I have referred to the same for preparation of this particular presentation. And there are some additional paper references. I have put up specific references with the topics. These are some additional references which will let you know about the entire process of development. Okay? So I would suggest that all of you go through these papers. Please do not rely only on books for your preparation. Read research papers also, because if you're going for a PhD, this is what is going to really help you out. And with this, I would like to conclude my talk. Thank you for such a patient listening. And in the beginning, I really had very good responses. Because we were short of time, I could not give you a chance to answer all questions, but I'm going to give you 20 more questions to be attempted, which I would wish everybody would really attempt. What is Mad Box? No, no, no. And it is not Mad Box, okay? It's Mads Box, M-A-D-S. Actually, we don't call it MADS, we call it MADS because people make it mad. So it's not mad. And fat box is nothing, okay? If you search for it on the internet also, you'll get it as MADS box, which is involved in floral identity. So I think I answered the question the student has asked. Okay, so all the best for your CSIR-NET preparations. For any queries related to this particular topic, you all can contact me at monishakay@nuv.ac.in. So thank you very much, and I would like to conclude my session. Over to you, moderator sir.
Yes, thank you so much, Monisha ma'am. It was indeed a very brief and very informative presentation. I think so. Ravi has asked, that is, can we get your presentation? Yes, Ravi, you'll be getting shortly through the mail. Okay. Apart from that, from the organizing committee, we are very much thankful to you, Monisha ma'am, for taking out uh, ample of time and enriching the students with the developmental biology of plants. I am definitely sure that the genes which you have talked is much more relevant, and the questions which are asked, you have attempted that also. Thank you so much, ma'am. Yeah. So just instruction to the students, that is, tomorrow please be sharp at 10, okay, for the next session, and till then we'll take your leave. Thank you so much.